Type 1 interferons are proteins in our immune system that are released by immune cells upon detection of a threat, typically a virus. When these proteins are released, they bind to specific receptors on the cell, which in turn activates a signaling pathway termed the JAK-STAT pathway. This pathway leads to the expression of genes responsible for antiviral and inflammatory responses, which then attack any invaders and prevent them from further infiltrating our cells. Generally, this process is effective in fighting off infections due to how strictly it's controlled. However, its dysregulation is implicated in systemic lupus erythematosus, commonly referred to as SLE. When someone has SLE, their type 1 interferon signaling pathways are overactive, meaning more type 1 interferons are produced than necessary. The excess interferons will bind to their receptors and activate the JAK-STAT pathway in such a way that the immune cells are overstimulated, so they overexpress the genes responsible for antiviral and inflammatory responses. This then leads to overinflammation and tissue damage. Immune cells, called B cells, can also be activated and produce proteins that attack the body's own cells in response to the excess amount of type 1 interferons. Thankfully, many strides have been made in modern medicine when it comes to treating SLE. One of the more notable drug therapies is anifrolumab, which is made of a protein that is part of the human immune system and has the ability to bind to a specific target. This drug alleviates SLE symptoms by binding to the receptor that type 1 interferons bind to, resulting in the interferons being unable to bind and activate the JAK-STAT pathway. This limits the activation of immune cells and will result in an overall decrease of inflammation and tissue damage.